the world. Okay. So coming down to malaria vaccines, and I mean, before
Ella,
that, I
you may
must
have
say,
to click, got it? Do you see that?
yes, I clicked it.
Unless you don't feel comfortable and you want to leave.
Unless you want to leave the meeting. And I also forgot to mention that Amanda Chung is volunteered to help facilitate discussion and questions. Would you prefer that we keep the discussion towards the end so you can go through your slides, or do you want to take questions during?
I think I'm very flexible. If there are any burning issues between the slides, please do feel it's fine for you to interject. Yes.
Okay, Amanda, are you okay keeping an eye on the chat and their hand raising?
Yep, I am doing that right now.
Okay.
Okay. So can I proceed?
Yes. Thank you.
Okay. So the slide, the vaccines, we know they are one of the most cost-effective public health intervention, and this, I mean, and responsible for actually saving millions of lives from infectious diseases across the globe. And a safe and effective vaccine for malaria would definitely be an added tool to already the tools that we have. However, developing this has been a challenging prospect, and it has taken many years, over 30 years at least, to reach where we are now. And some of these challenges, to address these challenges, the global malaria vaccine community came together to establish sort of goals and visions, and to identify activities that could actually address some of these challenges. So this led to malaria vaccine technology roadmap that outline a plan for how stakeholders can work together to accelerate the development of an effective malaria vaccine. So an initial goal, this was developed in 2006, an initial goal was to sort of develop a first generation malaria vaccine that has a protective efficacy of about 50% against severe disease and death, and at least last longer than 1 year. Then this was revised in 2013, and now the goal is developing a malaria vaccine with a protective efficacy of at least 75% against clinical malaria, and also which is suitable for the administration to appropriate at-risk groups in malaria endemic countries by 2030. And it also proposes a goal of developing a transmission blocking vaccine, which can reduce malaria transmission, and this can support elimination in various settings. However, we all know that developing malaria vaccine hasn't been easy. There are so many factors, such as sheer complexity of the parasite itself, having sort of the stages both in vector and in human, and at each of these stages sort of expressing different arrays of proteins. But also there's extensive antigenic variation for some of the very important antigens or proteins that have been the target of malaria vaccine. But the parasite itself also has redundant pathways to attack the parasite, making it a quite elusive parasite to control. Added to that, there's been sort of poor understanding of the immune correlates of protection. So the interaction between the malaria parasite and human immune system, sort of making it that this process being a very tedious process where we have to go up to the, to the field to really find out whether the vaccine works or not. And we haven't had a very robust correlate of protection other vaccines. So this has been these have been some of the challenges facing the development of a malaria vaccine. So in terms of the targets, I mean, there've been targets, different different stages, of course, and trying to block the development of parasites. And I mean, with well over 5,000 protein coding genes, we barely scratch the surface when it comes to finding the potential target. So some of the vaccine target the pre-parasitic stages of the vaccine before the parasite invade the liver cells. And this is an example here. It's an RTS S, which has been recently endorsed by WHO, but also others focus on erythrocytic stages, stopping the multiplication of the parasites, and transmission stages, blocking the transmission of parasite to mosquitoes, and development of parasites, transmissible parasite within the mosquito. So this has been some of the targets, and you can see some of the candidate vaccines there. And some of these are at different stages of development, with, of course, RTS S leading the way, becoming the first vaccine to be endorsed by WHO. So I'll go back and talk a bit about my work on phase 2 trials looking at RTS S. So when we did this work between 2008 to 2009, I focused on RTS S in phase 2 trials in Kenya and Tanzania, and but I was also, of course, involved in a phase 3 study that took place across 7 countries across Africa. So here in my work, I focused on describing the long-term outcome of the RTS S in children who receive RTS S. We looked at the analysis of waning efficacy over time and potential rebound malaria in those children who were vaccinated with RTS S. We also looked at the interaction between the malaria exposure and RTS S. We explored different models for the analysis of the vaccine efficacy. And finally, look at the correlate of immune protection and the association with the outcome in these vaccinees. So one of the things which became very important when we started working around RTS S, but also in immunology, in cohorts that were planned to look at the immunology, on understanding the malaria immunology, was the issue of malaria exposure. So we know that the heterogeneity of malaria, which is a very common phenomenon in the field, and this we know that could bias the estimates of malaria vaccine efficacy over time in longitudinal studies. It's quite sort of obvious that when you do, for instance, these studies estimating time to event, sort of using time to event analysis, if you have the heterogeneity in exposure over time, as more exposed and vulnerable population at risk are removed, there's increasingly less exposed and unexposed population left. And these may give an impression of the waning efficacy over time when you do this time to event analysis. But likewise, for the cohort studies that look at the effect of the immunity on the risk of malaria, again, if you include those individuals who are not exposed, this may wrongly give an impression that either the immunity is protective or not protective, depending on which group these individuals are included into. So I think all this led us to sort of focus on finding a better way of describing or characterizing the malaria exposure in an individual, so that we could actually adjust for it in our analysis for the vaccine trial. So we opted to work on this distant weight proportion of looking at sort of using an index individual and looking around a radius of about 1 kilometer and look at the prevalence of or the incidence of malaria or prevalence of malaria in the population around that index individual. And this was made possible because we had a lot of data. We had a lot of surveys going on in the study area, and this made it possible. And I'm sure if you don't have such kind of a platform, of course, it is difficult to develop such a measure. So what we did was to, of course, do a distant weight proportion for that, and this is described in this diagram here. So if you have 5 individuals and 2 of them are infected, a simple proportion will give you about 60% of individual around the radius are infected. But if you factor in the weight, giving more weight to individual closer to the index case, then that proportion goes down to about 22. So I can imagine we did that for all the population, for all the children participating in the study area, and we had it as a time variable, as time varying variable, sort of looking at 1 year and sort of 6-month blocks of time, and sort of adjust that that factor into our analysis of efficacy. So when we looked at the discriminatory power of these measures of malaria exposure, they're pretty good at discriminating between infected and non-infected children over a period of time, and we went ahead and utilized them in our analysis.
And sorry, what's the gold standard for those analyses to generate the ROC curves? Oh, to generate those ROC curves, what did you use as a gold standard?
Yeah. So what we did, we for each individual. So for the index, so we for this index child or index individual, we after calculating the distant local prevalence of malaria infection around 1 kilometer radius, and then we use a Poisson regression model to assess the discrimination power of this. So we looked at the prevalence, how much it predicted the incidence of malaria, both clinical and asymptomatic malaria in that index child. So the index child, we follow the index child and look at the prevalence of what was happening around, and sort of try to see whether that prevalence could actually explain or could determine the risk of an individual developing malaria over that period of time.
Yeah.
So that's what we did. Yeah. So so what we did was we sort of incorporated that measure into our analysis. But to also add, I think we also what we also did for a proportion of individual in that population, we did have data on antibody to blood stage antigens, and we also use those as well to describe or to characterize the exposure. And sort of when we put the two, I mean the exposed malaria exposure index plus the antibody titers to merozoic antigen, the model was more powerful in describing the incidence of malaria in that population. So definitely these antibodies have been used as a marker of exposure by various other groups as well. So what we did was then to use the measure, first of all, to in addition to adjusting for our estimates based on that, but also we categorized the population into areas of high malaria exposure and areas of low malaria exposure, and we looked at efficacy over a period of time. So this work, which was published in 2013, showed that over time, so the left graph is an area of low malaria exposure. Sorry, the left graph, yes, and the right graph is the area of high malaria exposure. And you could see that over time, and then of course they're divided based on the blue bars are the RTS S vaccinase, and the red bars are rabies vaccinase. So the y-axis is the malaria episode per person per year, so the incidence over time, and the graphs below represent the efficacy estimates. So if you can see that over time in areas of low malaria exposure, there was sort of less of waning of efficacy over time compared to areas of very high malaria exposure. And as you can see, in high malaria exposure, starting at around year 3 and year 4, we started to have more cases of malaria in children who were vaccinated. So this was enough to offset the advantage that we saw in earlier sort of years. Although at the end, if you consider all 7 years, it wasn't really statistically significant for sort of define the rebound of malaria. But we did see a rebound in terms of, from the 4th year onward, consistently we had more cases of malaria in children who were vaccinated with RTS S. And this was also seen in a phase 3. I think if you look at the data for severe malaria, there was that sort of rebound in later years. But again, the children were very much protected in the first 2 years, or we say 3 years of their lives after receiving the vaccine. So this was very important finding. I think it also informed the decision, or the analysis sort of approach in the phase 3 data. So we also looked at the correlates of immune protection. So we clearly showed the importance of antibody responses as of, to the protection that was seen or afforded by the RTS S. And we also demonstrated that, in fact, T cell responses were also contributing, as although to the lesser extent, to the efficacy that was seen. So you can see that in the top left graph, this is what we did. This is the analysis we did from the earlier data from malaria naive individuals. We analyzed the data and showed that antibodies were more sort of more determinant of the protection compared to the CD4 T cell, and this using different definition of malaria. But we also showed in a phase 2 that different individuals with the CD4 producing tumor necrotic factor alpha, those with higher antibody, sort of with the higher CD4 T cells, were protected compared to those with the lowest titers. But more remarkable finding was that you need definitely both antibodies as well as CD4 T cells, and when you have these together, it's very clear that the individuals are protected, and you can see by the lines. So individuals in a yellow line there in our KM plots, who had high antibody titers, but also higher CD4 T cells, most of them did not develop malaria over a period of 6 months of follow-up. So this sort of,
And Ali, which groups like didn't really have CD4 T cell responses? Is that did you have a lot of patients with HIV/AIDS, or this is just trends in a population that doesn't have HIV/AIDS?
So this was the phase 2 data. So we didn't. We actually excluded all those who were HIV positive. So these are HIV negative children aged between the 5 and 17 months. So the graphs, apart from the top left table, which came from the malaria naive population, all the other KM plots were data that were obtained from the children aged 5 to 17 months in Kenya, in Kilifi, Kenya, who participated in a phase 2 study. So all of these were HIV negative.
Okay, thanks.
So then, of course, the data from phase 2 were very promising. We demonstrated the efficacy of around 45% against chemical malaria, and that led to this large trial, phase 3 trial, which was done across 7 countries, which also, showed the efficacy of the vaccine to be around 36%, and the efficacy against severe malaria in that age group to be around 30%. There were very clear trend toward high efficacy in areas of low malaria transmission as compared to high malaria transmission, but more cases were averted from areas of high malaria transmission. This led to the pilot implementation of this vaccine in 2019 in 3 countries. Earlier, there were some safety concerns, and this was increased febrile seizure after vaccination. We saw increased risk of meningitis and cerebral malaria. But in a phase, in a pilot implementation, these, of course, were not observed. And the vaccine, of course, demonstrated 30% efficacy against, admission due to severe malaria. And it has been recommended for the areas of with moderate and high malaria transmission. And the benefit of the vaccine is on top of what is already being done in these countries. So meaning that with the use of bed nets, with the access to care, you still see this 30% efficacy in reducing admission of children with severe malaria. But this was only restricted, of course, in children the age of 5 and 17. What we do not know yet, and I think what is still the data being collected, is the benefit of the booster, because remember in a phase 3, it is the booster, the 4th dose, was very, very critical in maintaining the efficacy over time, as opposed to those children who received only 3 doses. So this is still to be determined, and of course, the impact of mortality is still being assessed until now. So another thing I wanted to mention, and which we are participating currently, is another CSP-based subunit vaccine. So this is another pre-erythrocytic malaria vaccine. It's called R21. It's very, very similar to RTS S. The construct is very similar to RTS S. Of course, there are more circumscribed protein antigens there in that viral-like particles, and there are no hepatitis B surface antigen no unbound hepatitis B surface antigen in the R21. So this is it has been shown to be highly immunogenic in malaria-naive adults, and impressive results were reported in Lancet a couple of months ago in Burkina Faso, where the vaccine was found to have an efficacy of about 77%, well above the 75 goal that was set by the WHO in terms of finding a malaria vaccine by year 2030. So this really created a lot of interest among malariologists, and it led to plans to conduct a phase 3 to validate these results across 4 countries. So Tanzania, Kenya, and Burkina Faso and Mali are participating. Over 5,000 children will be recruited. And in Tanzania, we have recruited 600 children already who have been vaccinated, and the vaccinations are ongoing. So it will be very interesting to see the results. After the RTS S has been recommended. So we all want to wait and see what will be the efficacy of this vaccine. So we'll know this, I think, by hopefully before mid-year this year. That's the plan to be able to share the data, at least with the WHO. So the other work that I have been involved, or the team here has been involved, is the sporozoite malaria vaccine. Of course, this is developed by Sanaria, Stephen Hoffman group, and this whole sporozoite malaria vaccine. They have 3 products that they're developing. Of course, is irradiated sporozoite. This is the PFS-PZ vaccine. We have the chemo attenuation one, so they use live parasites which can cause infection, disease, but they're attenuated with a drug which sort of clears the blood stage parasites, and they have a genetic attenuated parasite as well, which are under development, sort of genetically modifying the parasites so that they don't develop past the liver stage. The challenge, of course, is this has to be administered by intravenous administration, but we have demonstrated that this can be done in a research setting, and of course, more has to be done if this vaccine is found to be effective, sort of make that to train the healthcare workers in the rural, remote areas to do this. So it's always there'll always be a challenge there to be addressed if it is to be rolled in real setting. It was very efficacious in challenge study in malaria naive adults. Sort of the paper by Einstein showed very clearly that the 5 doses were very good at almost preventing all the infection following challenge. So the results in the malaria naive adults led to the studies in Africa, and in Tanzania we did the first study to look at the PFS-PZ vaccine before we went to another country called Equatorial Guinea, which is in the Central Africa. So the study in Central Africa, which was published in 2021, and that is sort of top right corner graph, it shows that in this study we compared all of sort of 2 of them, the irradiated PFS-PZ vaccine and a chemo attenuated vaccine. And the chemo attenuated vaccine, of course, had higher estimates of vaccine efficacy compared to irradiated sporozoites, and this was somehow expected because with the live PSPZ, this sporozoite sort of fully develop into and express more antigens, obviously, in the liver cell, which is critical for the induction of protective immune response. What we also showed in Equatorial Guinea and in Tanzania is that there was sort of a relationship between antibody responses as measured, these are anti-circumscribed protein, and the exposure or background transmission. So we saw, if you look at the graph the lower left graph, it shows that the individual in the USA had much higher antibody titers compared to, as you can see, far right individuals or others from Equatorial Guinea there. And we also, of course, demonstrated that infants sort of produce more antibodies responses compared to adults. And in Mali, our colleagues in Mali also showed the efficacy of the PFS-PZ in the field. So this was again measured against sort of natural exposure of around 20% there. So very much mimicking what we saw in the challenge study, despite this study in Mali being assessed against the natural parasite malaria exposure. All the challenge studies, just for your information, definitely they were homogeneous challenge. So the individual were challenged with the same strain of the parasite that makes the vaccine. So the other work.
Sorry, Ali, can you remind us with the chemo attenuation, what's the drug regimen?
So we have used chloroquine in Equatorial Guinea, but there are also other drugs that the team in Scenario are working on. But the experience, at least we have in Africa, has been with the use of chloroquine, which is not a very good drug to say the least, but that gave very good, gave the results that I shared with you here.
Mm-hmm. Thanks.
I think the key is, yes, the key is to sort of target the merozoites sort of which are released after the liver stage, and there've been a discussion of different drugs that could do that. So and not sort of target the Schizons within the liver stage, because those are the ones we need them to develop to the fullest to provide the sort of to induce the immune response.
And the timing of that, do you start before the vaccine or afterwards?
That's a good question. Yeah, so I think for the chloroquine, after the challenge, we do. So normally we will challenge the individuals, and for the sporozoite, it takes about 6 to 7 days before you start to see the parasites in the blood. So we would time it such a way that as soon as we challenge the individual, then we would start to give them chloroquine, and will not wait until the parasites are out. So we sort of immediately after the challenge, we also provide the chloroquine to clear any parasite that will come from the liver.
Mm-hmm.
So the blood stage vaccine, this is another area that we've been we are very active at the moment. We're working very closely with a group from University of Oxford. The RH5 vaccine is being developed by Simon Draper's group, and this is the reticulocyte binding protein homology 5, which is has been found to be very important in the pathogenesis of malaria, and play a very critical role in the invasion of the parasite, invasion of the red blood cell by the parasites. So what we are currently doing is to evaluate this vaccine. Of course, it has been developed for the target group is children who are at higher risk of malaria, and we are assessing the magnitude and quality of the immune responses and the longevity of these antibody responses, and we're trying to understand the specific epitopes which are critical for the inhibition of parasites. But we're also working with other two proteins, cysteine-rich protecting antigen and RH5 interacting protein. All these form complex together with the RH5, and as you can see down in the graph there, the antibody induced, and these were done in rats, the antibody induced in when both RIPI and CPRA plus RH5 are given are more potent in inhibiting parasite growth, meaning that if this is translated into clinical setting, we should expect to have a better protection with the three antigens together. So we are working, of course, we are now working with RH5, but I think the plan is also to go and evaluate the three antigens as part of the EDCTP funded consortium led by the colleagues from Burkina Faso. So this is also ongoing work. Again, RH5, unlike other merozoite antigen, is relatively highly conserved protein. As I said, it's essential for the invasion of the RBC, and it also induces high level of antibody, sort of strain transcending functional antibodies. It has been able to block most of the strains that have been tested so far. And also the important thing about this is that the assays, the growth inhibition assays, has been a very strong predictor of the protection, both in non-human primates, but also recently in a phase 1 study in UK, it has been shown to be quite a strong correlative mark of protection in humans as well. So our studies in Tanzania, they're looking at RH5 and CYRPA and VPI. We did the first study using viral vector expressing RH5, but now we are working on sort of protein adjuvant. So we're using matrix M as an adjuvant in Tanzanian adult and infants. But we are also working to establish blood stage control malaria infection. This is to support the efficacy studies. So we want to challenge later on adults and see whether we can demonstrate efficacy in our population in endemic country. Of course, we are building the capacity and infrastructure to support such kind of studies, which will be for the first time we're doing them in Africa, especially the blood stage challenge. So the viral vector trial using 2 viral vectors. So first we'll sort of prime you and then boost you. So we'll prime, we prime the individual with a CHARD63 expressing RH5, and then we boosted them 8 weeks later with the modified vaccinia Ankara. This is another virus expressing RH5 as well. And this was the randomized double-blind control trial. It was an age de-escalation, so we started with adults first and slowly went down to infants. And of course, we had 18 to 35 years, 1 to 6 years, so an intermediary group there, and infants 6 to 11 months. And the control vaccine here was rabies vaccine. So the manuscript for this work is under preparation now. But what we saw was, the vaccine was very safe and well tolerated by all age groups. What was very fascinating, of course, we observed this 15-fold increase in antibody in infants compared to adults, and because in blood stage vaccine, what is important is both the level of antibodies you can achieve, but equally important is the ability to inhibit parasite. And for the RH5, it has been shown that the ability to inhibit the parasite, of course, is a function of the antibody levels. So having showing that the antibody levels in infants is much higher than in adults is quite promising, because it means if we have good GIAs or growth inhibition assay results, it could really mean that this vaccine could have a potential of making a significant impact in the field. So we're all excited and we're waiting to see the final GIA result. We're still sort of cleaning the data up, and probably this will be published very, very soon.
Thanks, Ellie. This is Amanda. I'm keeping an eye on the clock, and I just want to be cognizant of giving some people an opportunity to ask questions. So if you could maybe wrap up relatively soon, but I also don't see any hands up or any questions in the chat just yet, but just wanted to give you a time check. Thanks.
Thank you very much. Yes, so I'll try to not rush, but quickly wrap up. So one of the other vaccine, of course, we're working on, as I said, is the adjuvant RH5 adjuvant in so the protein adjuvant, and this is ongoing, and we are recruiting about 90 participants here, and this is a different formulation. As I said, we are very keen, we are very interested in defining exposure. So we're also using antibodies to Scheizans to try to characterize the antibody responses, to characterize exposure, because this can actually modify immune responses and can affect the estimates, the efficacy estimates. So we are working to develop this. This is one of the work by a master's student, Tuono there, working with his colleague. We're also working on second generation transmission blocking vaccines. This is a one of the examples, the PFS 25, which has been combined with an immunomodulator IMX 1313, and this is hopes to improve the immunogenicity of PFS 25. And this is the phase 1 first in human trial ongoing in Bagamoyo at the moment. So this is the study in brief, I think, and we will be doing also standard membrane feeding assays and direct membrane feeding assays to actually measure the in vivo, ex vivo, efficacy of the vaccine. We're also working on the full-length MSP1 candidate malaria vaccine. Recent studies in Germany have shown that when you use the full-length MSP1 vaccine, perhaps you can include more T and B cell epitopes which have been actually left with the previous MSP1 candidate, and this could be very important in the immune response. So we're very excited, and we plan to do the phase 1B in Tanzania, including challenging these individuals very soon. As I said, that we are working on to establish a blood stage model in African adults and extend this to in vivo transmission model to be able to study the efficacy of the vaccines, blood stage, but also malaria transmission, malaria blocking transmission intervention. Last but not least, of course, we are very keen that we are working with the population. So we have a Wellcome Trust funding to define, the community engagement approaches, provide evidence of their importance. So we are working with the colleagues in Tanzania, locally here, and across Africa to generate this evidence, and of course, define the best practices when it comes to community engagement in clinical trials. And these are the pictures of the clinical trial facilities in Bagamoyo. This is a team, and this is some of the capacity building activities across Africa. We have been really involved in South-South collaboration as well as North-South collaboration, all aiming at building our capacity. And this is what we have been working most with University of Oxford, Swiss TPH, recently University of California, San Francisco, University of Global Health Equity in Rwanda, and Cambridge Wellcome Trust. Thank you very much.
Thank you so much, Ellie. We really appreciate you sharing your expertise and your findings. I am happy to moderate the question and answer session right now. If you would either put your question in the chat, or if you can raise your hand, then I can call on you.
Thank you so much. I'll just jump in. Thank you, Ali, for sharing this really impressive and important body of work on malaria vaccines. And it's really exciting time for the first malaria vaccine to now be recommended by WHO and to see all these other candidates in the pipeline. There's so many that you're not in the vaccine field. It's hard to stay on top of. Which ones do you think kind of are like the most promising, or maybe you can't say, but in terms of efficacy and feasibility issues, especially related to administration or production? Or do you think that some of these things don't really matter because later on you can create multivalent or vaccines that have multiple targets, and they can be modular, and you can just sort of add on other antigens or but it is quite hard to sort of switch to another vaccine or change policies, as we've seen with COVID. So some sort of like coordinated and streamlined approach. And I'm sure you've been in many discussions about this, but I think not everyone on this call is in those discussions. So it'd be interesting to hear your perspective about that.
Thank you very much, Michelle. It's true that there has to be some coordination here. Some of there's so many vaccines, as you said, and I think, I mean, the WHO, for instance, have always also emphasized on sort of work coordination among scientists and to make sure that we, I mean, the aim we all want is to have a very good, effective vaccine, and this is very important. And definitely RTS is not the last sort of like, it's sort of, our last effort. We still need to work and find much better vaccine. So maybe I might be biased, but I think still, investment in malaria vaccine field is still very important. And at the moment, of course, apart from RTS S, which has made with this milestone, which has really put the malaria vaccine in the spotlight, that was the first generation vaccine. So this second generation vaccine is coming up. The close one, which is now in phase 3, is R21. So the question is how different R21 is going to be from RTS S. Is anyone guess? Because they're very, very similar. Of course, the early studies in Burkina Faso have shown very amazing efficacy data, but I think it's too early to say anything until we see the phase 3 data, and this will come up, before next mid this year. Another approach which you've said well is this approach of combination, and of course there are technology people develop both on the technology side, sort of developing platforms where sort of you can add on and have different antigens on top of that. A good thing is messenger RNA. There are groups, of course, working into that as well, looking at whether you can have several antigens addressing different targets and sort of package them within the same platform as messenger RNA has been used for the COVID. But this is still at infancy. Malaria is not as any other disease we all know. It has been very difficult to develop vaccines, and we barely understand, what it takes to protect you. So it's going to be a challenge. But at least what we know for now, very important to have RTS S out there, and of course these other coming vaccines such as R21. PFSPZ vaccine, I would say there are challenges with the administration, as you rightly say, but again, if they can demonstrate a very good efficacy, I think it's a matter of having technologies and training to sort of making sure that it can be given to the remote areas of Africa where malaria is the biggest problem. Finally, yes, combination is important. There are groups working on that already now, and but again, initially these vaccines have to be evaluated individually before you can come and put them together. There are issues of interaction and sometimes synergism, but also they can conflict each other and have less immune response. So I think it's very complex, but I would say research is ongoing, and scientists, as you rightly say, there has to be some form of coordination, and this is happening at some level, at higher level, sure. And for me, the future is bright, but we need better vaccines for Africa. For someone in the field, we definitely need better vaccine. I've been asked, for example, I was part of the our national effort to evaluate these results from RTS S, and the National Malaria Control Program experts, they asked us, what do you think of this vaccine? And I think for us was, we have something now. We have to look at how we can use it, but definitely there will be better vaccine. And as a country, we have to be open. We have to sort of follow the development and see which one will be the best vaccine to come. But definitely we need to look for better vaccines. Hope.
Ali, we have 2 questions in the chat. I'm also cognizant that we're over time, so hopefully you don't have a hard stop, but please feel free to leave if you have other engagements. The first question is, though, how is COVID vaccine development and innovation informing malaria vaccine development? And then I'll just give you the second question as well. While I'm at it, the question is, what are your thoughts about the reasons for reduced RTS vaccine efficacy in higher transmission settings? and in young infants versus children?
Thank you. So for the COVID, thank you for the questions. Very good question. I think for the COVID vaccine development innovation, these are very important, I think. And as I said, there are efforts to develop a malaria vaccine using the platform. I think biotech had shown some interest to develop that, and there was some high-level discussion, of course, with all stakeholders to start, engaging with the stakeholders, both in Europe, the funders, but also countries in Africa, in putting together a consortium to evaluate this. So that is definitely ongoing. As far as the RTS S, the reason for reduced vaccine efficacy in higher transmission settings, I think it is. I mean, for me, I'm looking at it as in higher transmission. Definitely, in the first year of life, you can still avert many cases, but then there's sort of a quick waning. And I think this could be because sort of, the antibody responses you get in RTS S, they sort of have a peak and then they start to wane very fast. And with a higher force of infection, definitely some of these parasites are able to punch through your immune responses sort of barrier and you get infection. So I think it is because of both the antibody kinetics, so you have sort of peak and then it sort of start to fall down. And with a high level of transmission, this is sort of faster waning as compared to areas where there's no malaria, transmission is lower. But the last one is about infants and children. I think there are many sort of thoughts about that. One, of course, in infants was we felt perhaps maternal antibodies interference with the vaccine. The sort of immature immune response, but it's true that we didn't get very good result in infant, and this could be some of the factors that might have contributed over.
Great, thank you. I don't see any other questions in the chat. So once again, thank you so much for sharing your expertise and your time and your work with us. We really appreciate it.
Thank you
very much for having me.
And I'm sure we'll have more opportunities to interact in the future as we develop our collaborations. And if anyone wants to follow up with Ali directly too, you're welcome to. I think his email is on the invite, or we can provide it. Thank you, everyone. Thank you, Ali, for joining in the evening.
Thank you very much. Bye-bye.
This is lovely. Bye-bye. Thank you.